This figure has a substantial amount of information in it, so I'd like to take some time to discuss it with you. What we're looking at is a flow of information, starting with the DNA and ending with the polypeptide or the protein. We're not going to make a major distinction between these two categories. That can certainly be done, but for our purposes, when the mRNA is translated to produce the polypeptide, we would refer to that synonymously as the protein. But let's go back and look at this particular portion of the figure. Notice that on this side, the upper portion is the nucleus. Eukaryotic cells, of which human cells are, contain a nucleus. The region outside the nucleus, but inside the plasma membrane, is referred to as the cytoplasm. Now, within the nuclear membrane, or the nuclear envelope as it's called, there are pores, and things can fit through these pores to both leave the nucleus as well as to enter the nucleus. In this case, we're focusing on leaving the nucleus. So we're going to start with our DNA, and specifically, we're thinking about regions of DNA that have information to make things, and we would call those genes. So within the human genome, there are approximately 18,000 to 22,000 genes that are scattered across the chromosomes. These genes will undergo the process of transcription. And remember, that's the focus of this particular chapter. How is that information in DNA encoded into complementary RNA? The initial product of transcription is, in the case of mRNA, is referred to as the primary transcript. And that distinguishes it from the second form of RNA, which is sometimes called the mature mRNA. Later in this particular module or topic, we'll talk more about what distinguishes the primary transcript from the mature mRNA. The mature mRNA will then move through the pores and enter into the cytoplasm. In the cytoplasm, the second process of gene expression will take place, and that's translation. And translation involves the reading of the mRNA via the ribosome and the use of amino acids to create a specific sequence of amino acids. And that's referred to as the primary structure of the polypeptide. Remember, the protein can then twist and fold on itself to take on a more complex folding that would be associated with its final form. In other words, what's referred to as here as the finished protein. In a nutshell, this figure is describing the process of gene expression. The first process is transcription to produce RNA. If the particular gene specifies the production of mRNA, then a second process will take place in the nucleus, which is called RNA processing. And in the reading, you'll see the 3 steps to that process: the addition of a 5' cap, the addition of a 3' poly A tail, and the removal of introns and the splicing together of exons. That's what gives us the mature mRNA. The mature mRNA will then leave the nucleus, undergo the second step, which is translation to produce that polypeptide. So I hope this video is helpful in deciphering the tremendous amount of information that's included in this particular figure.